Great. Hi, everybody. Welcome to our SAAB webinar. We have a great evening set up for you. And then I'd just like to remind everybody, if you have received an email from Elizabeth about next year's SAAB conference, to please fill that out or remind your supervisor to fill it out so that we get an idea of what's going to happen. And with that, I'd like to hand it over to Anina to introduce our speaker for the evening. Thanks, Anina. And thanks everybody for coming.
Thank you very much. Yes, welcome everybody. It's really great to have a nice audience here tonight, virtually. And it's such a privilege for us to showcase the interesting and wonderful work that the SAAB community is involved with. So just before I start with all the administrative stuff, remember to add your name into the chat if you want to receive SACNAS points for attending this webinar. And while we're busy with the webinar, please keep your microphones and cameras off so as not to distract. But you're welcome to ask questions at the end. So you'll see that there's a chat function there where you can add any questions throughout the webinar. But please also, you're welcome to ask questions at the end, which you can do through the chat function or just by raising a hand and asking questions. So we look forward to that. So tonight, we have our SAAB bronze medalist winner to show us about her work. So this award is given annually to the best PhD in the field of botany, so in a range of botanical fields which are judged. So it's really a great honor, and it's we're very excited to see what you've got to present here. And in general, it's just nice to see what the young botanists are up to these days. So we're really excited to see this work, Hannah. Thank you very much for that. So Hannah's just going to tell us a bit more about herself. She's prepared that in the talk as well, so that's great. So you can get a bit know a bit more about her background, and then she'll go ahead to tell us about what she's been doing. So yeah, I think that's all that I need to share with you. So please enjoy the presentation and remember to ask questions at the end. Thank you very much.
All right. Hi, everyone. I'm Hannah, and today I'll be telling you a little bit about my work that I did during my PhD, as well as my postdoc. So just to introduce myself, first of all, I'd like to say thank you so much for having me. To do a SAAB webinar is a very cool thing to be able to do. So I'm currently a postdoc at Rose University with Dr. Ethan Newman. And I did my PhD in botany at UKZN with Professor Steve Johnson. And with both of those supervisors, I've been looking often at pollination, but also some work on seed dispersal. And I'm particularly interested in how ecology drives evolution. So today what I'll be talking about is specifically how this works and how ecology, for example, pollinators can drive evolution and speciation. So if we look at this photo, or if you go outside, quite frankly, although maybe not at the moment in winter, you'll see this amazing floral diversity around you. And we have this quite a wide range of different families of plants in the angiosperms that have flowers. And these flowers vary dramatically in shape, size, color, scent, etc. And a question that I have always been interested in, to be honest, since I was a little kid, is why is there such floral diversity? Why do we have this massive amount of different types of flowers? And one of the key answers to this is pollinators, and that's what I'm going to be talking about today. So how does speciation work? So just the basic understanding of evolution, right? How does a species become a species? So gene flow barriers will evolve within or between populations, in this case of plants, right? And these barriers can be ecological or adaptive, okay? So for an example, we can look at Keeble and Johnson's work on clivias, and we see that there's different species here, and there's a butterfly pollinated species and 4 sunbird pollinated species. And in this case, the gene flow barrier is ecological because it's the butterfly species that has resulted in a new species of clivia as a result from this shift in pollination. So pollinators can drive evolution. And to break this down, a plant's purpose is to successfully reproduce. And a plant has a flower because a flower needs to create and receive quality pollen. And one strategy of reproduction is spreading your pollen around. And you can do this by using a pollinator. In particular, you can use a biotic pollinator. And therefore, a flower's purpose is not only to make and receive pollen, but it is also to attract a pollinator. And this is kind of the key answer to that question I posed. Why do we have so many different types of flowers? They're essentially there to attract many different types of pollinators in many different types of ways. So when we measure flowers, we are also measuring that interaction between a flower and its pollinator. So we're measuring ecology, and we're also measuring evolution. And I'll show how this really amazing little sentence has helped me in my work and in understanding the ecology and evolution around us. So floral traits essentially function to increase reproductive output, and a flower's morphology will evolve toward a pollinator's morphology. As an example, we have this rather dramatic example that Darwin saw with Angraecum piscopedala. So this is Madagascan star orchid, and it's pollinated by hawk moths with very long tongues. And it has a very long floral tube. And this is called a mechanical fit. So this fits really well with the long tongue of that hawk moth. And in this case, it's actually coevolution. So as the hawk moth's tongue gets longer in order to probe the long floral tube, the floral tube will get longer, which makes the tongue get longer, which makes the tube get longer, etc., etc. But basically, the morphology of one is being driven by the morphology of the other. So what is a pollination syndrome? This is something that I put in my title, something I'm going to be talking a lot about today. And essentially, it's the idea of the convergent expression of floral traits in unrelated species. And as an example, I just talked about how there was that fit between the long-tongued hawk moth and the long floral tube. Well, the long floral tube is therefore really important in hawk moth pollinated flowers. But it's not only that. That orchid that I showed is also white, and it produces a scent at night. And these 3 traits in particular are really key in attracting hawk moths. And so we can say that having a long tube, being white and being scented, is a pollination syndrome for hawk moths. And this kind of follows on to a very basic idea, which I'm going to try and show or explode onto you, I suppose, that can be wrong and can be right. But the pollination syndrome idea is kind of based on this idea of a 1:1 ratio. So one flowering species has one pollinator group. So if it's a 1:1, then this is basically specialization, okay? So here's another example of a pollination syndrome with very specialized interactions. So here is a South African example. We have Redivivia beringwii bee in the middle there. And around it are all of these flowers, these orchids, flowers that we see in the Cape. And all of them, just by looking at them, you can tell that they look similar. They have the same kind of coloration, and they're quite small flowers. But they also produce oil and have a pungent scent, and they all flower in September. So all of these morphologies of the flower have been driven by that bee species, so that we have a syndrome because we have a single pollinator that is associated with a range of flowering species that are all similar. And that kind of sums up what a pollination syndrome is. Interestingly enough, if we look at this picture, you can see at the bottom something that may look a bit more familiar to us, but perhaps I certainly haven't. Perhaps you haven't seen this particular color morph of Disperus capensis. And normally when we see Disperus capensis, the most common morph would be this pink one, which I've shown here. And this is a completely different pollinator, carpenter bees. And so in this case, we're already getting divergence. We're getting a shift from pollinators within the same species toward different pollinators. So even diverging from that pollination syndrome to another potential pollination syndrome. So I was really interested in this idea of pollination syndromes and in specialization. And so I work mostly in the family Amaryllidaceae. And people always ask me why, because it's pretty much all I've done, and I am very keen on them. And I think it's primarily because they're absolutely stunning to look at. There's a variety of floral morphologies that occur in a variety of habitats around the world. There's some key phylogenies that have been done, but there's not enough ecology research done. And to be honest, I think this is partly just because it is such a large family with such a variety of floral morphologies. In South Africa, the Amaryllidaceae, we have several genera representing several subfamilies. And here the diversity is astonishing in terms of flowers as well as the fruits. And this occurs throughout the country in a variety of different habitats, from wetlands to desert. Again, there's some research, and some of this research gives hypotheses for various other pollinators of different species. But again, because it's quite a large group, there's not always a solid amount of data that is to back it up. And of course, we continue to grow in our understanding of ecology and evolution, and we can always ask more questions about systems that were documented in the past. So what I'm going to do is I'm going to be showing the traditional syndrome on one side, and on the other side, I'm showing the species that I worked on, and I'm going to try and contrast them. So for example, we have a butterfly pollination syndrome of Pentanisia. So this particular species fits into the butterfly pollination syndrome absolutely perfectly. It has a thin little tube that fits that tongue of the butterfly perfectly. It has nectar as a reward. It's blue in color. It has a landing platform so that the butterfly can perch as it puts its tongue down the tube. For Scodoxus multiflorus, on the other hand, the species that I worked on, it's bright red, as you can see. It has kind of a tube, but it's also got really exerted styles and stamens. But these were also visited extensively by butterflies, despite not looking like they were part of the traditional butterfly syndrome. And this was partly because it was pollinated by butterfly wings. So we can see here on the image, Scodoxus multiflorus pollen. So that's those little yellow bits on the white wing, butterfly wing in the background. And what happens is that the butterfly will come in to drink the nectar. As I said, there's no landing platform with this species. And so it has to flutter its wings. And as it does that, it comes into contact with the flower, with those exerted styles and stamens, and in doing so pollinates the flowers. And when we look at other South African Amaryllidaceae, there's quite a few different species in different genera that look quite similar. So over here, A is Scodoxus multiflorus, just to compare. And all of these are, so we have Clivia, we have Nerines, and we have Sotanthus. And all of them look quite similar. And this is all because there are, at least potentially, hypothesized to be, and some of them have proven to be, but they're all pollinated via this particular butterfly wing system. So can we call this a pollination syndrome? It's different from the traditional butterfly syndrome. Can we call it a whole syndrome in and of itself? How big is the syndrome? Does it occur outside of South Africa? Does it occur outside of Amaryllidaceae? There's lots more questions that we can ask about this system. Right, onto sunbirds. I looked at Scodoxus puniceus. So the traditional syndrome we can take an aloe as an example. And again, it has those tubes for the sunbird's beak, and it has nectar as a reward. They're fairly sturdy flowers, of course. We're talking about a larger sunbird at this point. And it has what we call a perch, or somewhere that they can grip onto while they feed. Scodoxus puniceus, again, appears to not have a perch. It has hardly any of a tube in an individual flower, and strongly exerted stamens and styles again. And it's in this kind of what we call a paintbrush inflorescence that is surrounded by these bracts. So the idea of this is what actually happens, and we can see this in the video, is that the sunbirds come along, they can perch on top of those inflorescences, so they use the inflorescence itself as a perch, and they can then insert their beak to get nectar at the bottom of the flowers. And in doing so, they pollinate the plant. And all of these videos are from different sites, and they show different species of sunbirds. We also found that sunbirds were really important for the genus Hamanthus. And we looked at mainly 3 species: Hamanthus deformis, Hamanthus humilis subspecies hirsutus, and Hamanthus coccineus. And what's cool about Hamanthus is that it has quite a wide distribution range, and it occurs in kind of 3 color forms. And we studied these 3 species in order to make sure that we studied these 3 color forms. So coccineus, those from the Cape will know this species quite well. It occurs on the beaches around the Western Cape, and it's bright red. Whereas Hamanthus deformis occurs in small forested patches and savannas in the east in KZN. But both of these species are pollinated by sunbirds. What's interesting about Hamanthus deformis in particular is the fact that the inflorescence actually sits on the ground. And this means that the sunbird uses the ground as a perch and can then insert its beak from there. So here again, it's just a video showing. We also have Hamanthus albiflorus, which after setting up several camera traps over quite a long period of time, we got one perfect video, which is this one. And if you look carefully at some of the videos, you can see the amount of yellow pollen that's surrounding the face of that bird, and there's just perfect contact resulting in pollination. So for the humilis subspecies hirsutus, you can see it's slightly more pink in color. And some experiments showed that potentially other insects are important, but sunbirds are pretty important in the pollination of that species as well. So pollination syndromes, let's come back to that. Why do we have them? Okay, so we assign them, and occasionally it is effective. So as an example, if we look at the Balsaminaceae here, you can see that the different pollinators are associated with different floral types. So it is important to be able to assign a pollinator to a pollination type. However, I think this is a simplification of evolution. And importantly, many flowers do not fall so neatly into a syndrome. So how wide should this syndrome be in order to incorporate more? Do we need new syndromes? These are, many questions I keep in my brain. So coming back to the specialization question again, is everything specialized? Everything I've shown you, we have this idea that a specific pollinator has driven a specific flower. But actually, there's a debate as to whether generalization or specialization is more important. I've just highlighted 2 key papers from my field. And this kind of kickstarted in the 1990s, and the debate is still going strong. And I have some ideas about that. So what is generalization? Well, generalization is when a particular flower species has multiple effective pollinators, and proving that effectiveness is really important. So for example, we have Disofragrans here, and this is pollinated by at least 4 insect orders. You can see here there are flies, there are bees, there are beetles. I've asked this question, how specialized is generalization exactly for this species, right? looks pretty generalized, but it's still only insects. It's not anything larger. And then for dyes of fragrance, the scent is really strong, but it consists of many compounds that are known to attract many different insects. Can we have a generalized pollination syndrome? And this is the question I haven't seen anyone else post, so I'm putting myself out on a limb here. But I think it would be really interesting to explore the idea of a generalized pollination system. For example, where we have very particular traits that are actually associated with a generalized pollination system, such as a particular type of scent with certain compounds that are known to attract many different pollinators. Quite normally, though, we have specialization despite generalization. And this is because many plants are visited by a range of visitors, but only a few of them are effective pollinators. So as an example, we have Hypoestes aristata here. And as you can see in the image, they are visited by quite a large range, from larger sunbirds, different types of bees and flies. However, when they compared the seed set resulting from the different visitors, the highest seed set was from larger bee pollinators. And so actually these larger bee pollinators are the most effective pollinators, and these are acting as the only selective agents in this. So these are the things that are driving evolution for the species, even though it's visited by a range of visitors. We also get something very intriguing, which I've gotten into really recently, is the idea of multiple specializations. And this is the evolution for traits for multiple specific pollinators. So as an example, if we look at Adansonia digitata, so this is the baobab, the iconic baobab tree. And if we look at the flowers, we may immediately think, if you're a pollination biologist, you may think bats and be done with it. And this is because it produces a lot of nectar. It's a large, robust flower that bats can really hold on to and not necessarily destroy. And it's got that kind of head of stamens and styles, again, quite often associated with bat pollination. And for a long time, it was thought to be exclusively pollinated by bats. However, in many areas where the baobab occurs, it is in fact pollinated, and very effectively, by hawk moths as well. And at first, you might not think that this is a good fit and that the hawk moths are in fact cheaters. They're just coming along and the bats are the real pollinators. But actually, this flower has developed a scent profile that has become adapted to both of these at the same time. So although we may think of it as a generous species, it is pollinated by more than one thing at the same time, it's actually two very specific pollinators. And can we say that this is a multiple pollination syndrome acting together, right? Can we say that we have a bat and a hawk moth pollination syndrome together within the same one to create a new syndrome? In order for this plant to be pollinated by two effective pollinators in this case. So continuing with moths, firstly, let's look at the traditional moth syndrome in Crinum variabile. I've talked a bit about it. Remember that orchid example of Angraecum sesquipedale. And Crinum variabile is absolutely perfect when we think of what a hawk moth pollination syndrome would look like. And this is because it has a really long tube and really big flowers, and these flowers are white to pink in color, and they have a strong scent at night. And Manning and Sneeman showed that they were, in fact, pollinated by hawk moths. The species that we looked at, Crinum campanulatum, on the other hand, looks a bit different. So this is a near-threatened species, so it's only found in a handful of ponds around Makanda in the Eastern Cape here. And it has much smaller flowers and a slightly shorter tube. It produces less scent, and it seems to be during both the day and the night. And it's a darker color as well. So what did we see as the pollinators? Well, of course, we expected there to be large hawk moths involved, but in fact, we didn't see any. We didn't see any hawk moths at the sites either. We did light traps. There weren't any hawk moths visiting. And we didn't see any of them visit the flowers. So what we did see was small noctuid moths in the evenings. And these just didn't do a great fit. I've tried to kind of draw them to show a fit between them. And the smaller noctuid moths just didn't fit very well, and they didn't seem to carry a lot of pollen. Carpenter bees, bit bigger, very good fits. They come in, they drink the nectar that wells up the tube, and they seem to be covered in pollen. But the most abundant pollinators were, in fact, the honeybees. And these try to get nectar and they try to collect pollen. But the sheer abundance of them meant that they were a potential really important pollinator. So what we did is that we bagged the species either during the night or during the day, so that we could compare nocturnal versus diurnal pollinators and see which one resulted in the highest fruit and seed set. And actually, the day treatment had a higher fecundity than the night treatment, which meant that those diurnal bee pollinators are actually incredibly important pollinators. And those smaller moths are pretty negligible. They didn't actually have much of an effect at all. And the species, we did a couple other treatments, and the species is in fact not pollen limited. It should be noted that watching, we found both of these similar results over 2 seasons. So this has a huge implication for the evolution of the species. Interestingly enough, there was more scent produced during the day than during the night, although the compounds were similar. So this could be an example of intermediate stage of double function, which basically means that we have this initial one species' ancestor that is pollinated by hawk moths, and potentially we're in the middle of a shift toward a different pollinator of diurnal bees. And we are somewhere in the middle of that, where it can now be probably successfully still be pollinated by those hawk moths, as well as successfully pollinated by honeybees. And therefore it can accommodate both at the same time and still be effectively pollinated by both. But it could be on its way to shifting toward the bee pollinators rather than the hawk moth pollinators. Or again, is this an example of the multiple specialization hypotheses that I had before, of where we have both bees and moths as the multiple pollinators in this example? So I became really interested in looking at kind of evolution on the ground. How is it happening right now? As I mentioned all the way back in the beginning, this idea of measuring a flower and then measuring the evolutionary action that happens. So we looked at, for some of this, in Brunsvigia gregaria. So this is an absolutely stunning species that we find, a candelabra lily, that we find in the Eastern Cape here. And there are 3 ecotypes, which turn out to be kind of different in a PCA. So you can tell the differences between them. So there's a short-styled form. So this is a slightly smaller flower, and they're a darker pink color as well. There's an intermediate styled form, as well as a long styled form, and that one obviously has the longer style. And these are the visitors that we found. So the short was visited, and these fit quite well. You can see a honeybee, for example, there. The intermediate form, those larger butterflies were a pretty good fit. Whereas for the long-styled form, the only thing that would make a good fit were the long-tongued flies that visited it. So if we have a look at this diagram here, this shows interactions between the pollinator groups on the one side, and on the other side we have plant populations. And then the thickness of the lines are showing visitation rate. So at first, if we have a look at this, there are lines going everywhere, and it kind of looks like it's all over the show. But actually, once you start zooming in, you can tell that some of it is a bit more specialized. For example, if we look at the long-tongued flies, the LTFs, they're right at the bottom here, they only visit 2 populations, and both of these populations are the long-styled form. So actually, what is driving the evolution of the long-styled form are those long-tongued flies, because they're not in the localities of the intermediate and short-styled forms. However, the intermediate and short-styled forms are more visited by bees, and swallowtails here are more interested in those intermediate populations and very occasionally visit the long-styled forms. So then what we did is we came up with this very complicated diagram. So what we did is that we measured initial floral traits of an inflorescence, and then we counted the fruit and seed set of that inflorescence, again for the 3 different ecotypes, and we did a lot of these, about 100 per ecotype. And we can then get this graph where we have fitness SIFs for each population. And in this case, the yellow, so for example, in the intermediate, you have this bright yellow patch, and that is showing the highest fitness. So this means the highest number of fruit and seed set. And then going darker and darker, you'll have the low fitness. So we know that style length is really important. This is showing tepal and style length, but we know that style length is important in understanding these ecotypes. That's how we based our understanding of the ecotypes to begin with. And if we look at the evolution, so this is over 1 generation, what exactly are these pollinators selecting for? The short population is getting longer, the intermediate is getting shorter, and the long is not changing. And basically, the main take-home message from that is that evolution is an ongoing process. So it's not stable all the time. Even within this 1 generation, we're seeing beginnings of shifts and changes. Now, initially, we could have said that this was a generalist species. But once we start going down to it, and we start looking at those very specific ecotypes, and we start looking at what are the visitors and how are they affecting evolution, we can see that they're actually specialized ecotypes within a generalist species. So now I've put up a picture of alien plants just to confuse you, but essentially I'm thinking about current and future perspectives here. So for pollination syndromes, we have to remember that this is a concept and it's an educated guess. So once we assign a pollination syndrome, we have to double check this. It's a hypothesis to be tested. Direct observations are incredibly important in this. We cannot just assign something and hope that that's right, but we can use more refined techniques such as 3D modeling of flowers. And also, we need to remember that specialization can still look different from an expected syndrome, and this doesn't take away the importance of it being specialized. And then, like I've mentioned before, how do we set up a boundary on a syndrome? And at what point do we call it a new syndrome? At what point do we stop calling them new syndromes and settle with the ones we've already got? And at what point do we abolish pollination syndromes? And at what point do we keep them? I think we need to keep them, but I think we need to really remember that this is an educated guess rather than a blind truth. So specialization has long been recognized as important. Generalization does appear to be important, but I still have questions about what exactly is it, especially in terms of when we're talking about pollination syndromes. Is this multiple syndromes acting at the same time? Is it its own specific pollination syndrome? Does the pollination syndrome just not work for generalization? And so I have said that the continuum, I think, is the most important thing in our new understanding. I don't think we need to be thinking about specialization versus generalization. We need to be thinking about it together as a continuum, so that on one side we might have extreme specialization and extreme generalization on the other side, but it's more likely that there's a whole lot of plants that we are just not thinking about properly because they don't quite fit neatly into either box. So some questions from Agnes Delinger in 2020. What is the ancestral pollination syndrome? How often have shifts in a pollination syndrome occurred? Is there a recurring shift directionality? Are there any reversals? Are some syndromes more likely to shift than others? And we can answer some of these questions using what we call phylogenies. So we come back to that classic Clivier example, where we have one butterfly pollinated species and 4 that are pollinated by sunbirds. So in this case, the ancestral pollination syndrome was sunbirds. The directionality was from sunbird to butterfly, and it looks like this may have been the only one in the genus. So phylogenies show how things are related. And we can kind of call this the tree of life, okay? So phylogenetic analyses with state reconstruction can show what the past looked like, and therefore we know what has evolved since then. So as an example, let's have a look at the genus Dizer, an absolutely beautiful orchid species that occurs in Africa. And here we have the red species are nectar-producing species, so they have a reward for their pollinators, whereas the gray do not have a reward, something that's unique amongst orchids often. And if we trace it back, we can see that the ancestor was always a cheat. So this genus has been a cheat for a long time. It didn't have any reward for the pollinators, no nectar or anything. And therefore, nectar has actually evolved since then. And this nectar is an incredibly important way of showing how pollinators have therefore contributed toward the evolution of the genus, because they have selected for nectar, for example. But pollination studies can also be important in phylogenies. When someone makes a phylogeny, it can be kind of meaningless without an ecology, because we need to know why. Why are these species related in this way? Why are they not? Why is this a new species and this isn't? And when we just look at flowers, pollination studies will inform how and why certain flowers have evolved in the way that they have. So as an example, we look at the Dizer phylogeny again, these beautiful drawings by Steve Johnson. And I've put in here an example of Dizer uniflora, which if anyone's been around Table Mountain, hopefully you've had the chance to see it, or at least see some beautiful photos of it. And this is pollinated by the mountain pride butterfly. And if you look around this phylogeny, you can see that the mountain pride butterfly does in fact pollinate something else, but it's all the way over here. They're not related at all. And the Dizer uniflora is actually quite a large flower as compared to the rest of it, and certainly as compared to the more closely related species, which are not pollinated by butterflies. And so it may be that in this case, potentially butterflies have been really important in selecting for those large red flowers that we see in Dizer uniflora. And this may have caused or certainly helped in a part of the evolution of the flower and the species. So in summary, to put it all together, Amyloideaceae are amazing and everyone should be studying them. Nature is complicated. What we know is not enough. Field work is important. Whatever other people tell you about working in the lab, field work is important. And phylogeny seem to be the way forward if we want to truly understand evolution and how pollinators affect evolution. And this just means that we all have work to do and we have to get out and do it. Thank you so much for listening to my talk. Yeah, here are some of my publications, and thanks again for giving me this opportunity. I hope you all enjoyed.
Fantastic. Thank you so much, Anna. That was great. Definitely, I share your sentiment that there's a lot of field work that still needs to be done. There's so much fundamental interactions and biological things that we need to find out still. So I see there are some questions in the chat, so I'm going to start with that. But the audience is also welcome to indicate if they've got questions that they'd like to ask, either by raising a hand or just putting a note in the chat. So I'm going to start with some of these questions. So Matthew Moore asks about the sunbird pollination of Scadoxus punicis and Hemantha species. Is the pollen transfer primarily on the head of the bird? Does using the inflorescence itself as a landing platform allow for pollen to be transferred by the bird's feet?
This is a very cool question. because I found it absolutely fascinating when I was working on it, because you can see that there must be pollen deposition on their feet, but you can also tell in those videos that there's a lot of pollen deposited on the beak and the feathers around the top of the beak. And it may be that that is where more pollen is deposited, because when they land, they tend to grip and stay in one spot. But those feet could still be important, and it would be pretty cool if we could have captured some birds and tested for pollen. So I don't know exactly, but there are hypotheses that yes, it could be that feet are important.
Cool. And then Ruth asks, it looks like those moths on the crinum are Helicoverpa armigera, which are often day active. So could this strong daytime scent also serve a role to attract moths?
That's really cool. Thank you so much, Ruth. I do not know my moths at all, and so I've been wondering what this is. We saw them in the evening only. We never saw any moths in the full on day, but we did see them in the very early evening, which I hadn't always expected to see them sort of 6 p.m. around about, which in summer is still fairly early for moths. So I need to write that down and look it up. But that's very interesting. Yeah, thanks, Ruth.
Great. And then Ruth also asked, with regard to Brunsvigia gregaria, did you assess pollinator community independent plant species to infer absence of specific pollinators?
We did not. And I agree that that would have been a really important point. And again, anecdotal evidence could be that while watching for flowers, for example, the long-tongued flies were only present in the populations where there were long style forms. But it was tricky to kind of tell how are we going to tell the pollinator community. So we just looked at the flowers and what were visiting the flowers.
Okay, and then Graham asks, do you think the taste of nectar between plants could have an influence on pollination syndrome? Like one plant has more sugar and nutrients compared to another plant?
Definitely. It's something that I decided not to dwell in, because there's this actually, I saw a paper today that was published today that was very interesting. And there's a lot of stuff that has been doing research into very specifics of the types of sugars even that are related to certain pollinators. And I'm sure that as we delve further into understanding nectar, we can see how important that is, because you can certainly tell, again, from looking at the nectar, we can tell what the potential pollinator is going to be. So yes, nectar certainly forms a part of that pollination syndrome.
Great, thanks. And then Glynis asks, or she mentions that the outgroup or sister group choice will always influence the interpretation of trait optimization. So could this have influenced the lack of nectar in the dicer phylogeny?
I mean, good point. I didn't do the dicer phylogeny, so maybe chat with Steve. I would hope they did the right thing. I'm busy doing a similar study and trying to find out what is the perfect outgroup. Is quite fun in and of itself.
Okay, thanks. I was curious about whether you've recorded any nectar robbers in the Heymanthus and the crinum species.
Yeah. So for the Heymanthus and Scodoxus, they were often in some localities, they're absolutely covered in bees and honeybees. And they definitely seem to take pollen as well as nectar. And we did a caging experiment where only bees were allowed in and the sunbirds were not allowed. And we compared that to where everything is allowed to visit, which was much higher. And so we know that those sunbirds are actually the more important pollinators, and that the bees are potentially, those nectar robbers. For crinums, I think those smaller moths, again, if you look at those photos and if you look at the way that they're interacting, they go for the nectar. And I mean, honestly, those photos are perfect because it's almost like they purposefully avoid the stamens and the styles. Yeah, so I would assume that they would be nectar robbers. Yeah, they certainly, from the small percentage that we did catch, they didn't carry any pollen.
All right, thanks. I see Steve is here. Please, Steve, go ahead.
This is more by way of a comment, but in the discussion of multiple specialization, I think it's worth mentioning the idea of a bimodal pollination system, which is, I might be wrong, but I think it may be a term that Manning and Goldblatt came up with, I think describing Tritoniopsis species that were visited both by birds and mountain pride butterflies. So this is the idea that you could have a plant species that is kind of highly specialized, but for two very different kind of animal groups at the same time and in some kind of stable fashion. So it's not it's a bit different from Stebbins' kind of intermediate stage of double function. It's more suggesting that you might get a kind of a stable state, and I think it could be explored further. I mean, are those flowers sort of phenotypically in between the two different syndromes? Or is it something unique? And it also suggests that specialization, you kind of don't always have trade-offs. You can be sort of doubly specialized in a way, but not. It only works for certain types, like wasps and beetles often kind of go together, but some combinations don't work together. Or you can't have a very long-tongued insect and then something that doesn't have a long tongue. But yeah, I think it's something worth mentioning as part of the general debate.
Cool, thank you.
Definitely, thank you very much for that.
And just if I could just comment in relation to Glynis's question, yes, absolutely, outgroup selection can make a difference, but the whole of the orchids are ancestrally deceptive. So it's quite deep in the lineage that plants were deceptive. But the general issue of outgroup selection does apply. Yeah.
Thank you very much for that. Glynis also says thank you. All right, I'm just checking if there's any hands that I'm missing. I don't know if I missed it, but how often do you have multiple Scadopsis or Haemanthus species co-occurring in the same site? And could co-occurrence be a driver of pollinator shifts, or do they generally occur alone?
They generally occur alone. There's not so much overlap, and some of them are quite specialized for their quite unique habitat. I mean, like I said, with Haemanthus, right, you get the red ones to the west, and then you get the white ones to the east, and you get albifloss that are in forests and Deformis that are in savannas. And so you often get this quite specific to their environment as well. So the extent to which pollinators-driven speciation would be in question, but they've certainly driven the flowers' evolution.
All right, thank you very much. Okay, are there any other questions from the audience? All right, I don't see any questions in the chat and I don't see any hands up. I hope I didn't miss anything. But I think there was some really great discussion, some things to definitely for us to think about and chew on. We seem to be finding ourselves grappling with that question in forest pollination as well. When is it generalist, and to what extent that whole continuum is keeping us busy. So thank you very much for stimulating this discussion. I think it's really great, and I look forward to all the field work that we're going to do and to figure out more of these puzzles. So thank you very much, Anna, for your time and for this beautiful presentation. And all the best with your postdoc and all the other projects that you're taking on. I see some nice messages in the chats as well, so you're welcome to go back and look at those. And thank you very much. Yeah, sorry, a final word from you?
Thank you.
Yes, great.
This was absolutely great. And Ruth, message me the name of that moth. I need to make sure I know it, because that sounds very important.
Okay. Great. Thank you very much, Anna. And thank you very much, everybody, for attending. And I'm sure you all enjoyed it. Our next webinar will be in August, on 21 August, by our famous geobotanist in the SAAB community. So please look out for the adverts for that as well. So thank you very much, everybody, and hope you have a great evening further.